It is 2:13 a.m. The laboratory is completely silent, save for the dull, relentless hum of the ultra-low temperature freezers. The fluorescent lights overhead buzz like tired insects, casting long shadows across the stainless steel benches. In my gloved hands, I am holding a small, frosted vial. By every known rule of biology, the substance inside this glass should not be able to frighten a grown scientist. It contains no DNA. It contains no RNA. It has no genome, no genetic code, no blueprint for life whatsoever. And yet, if this substance were to enter your body, it could spread like a rumor. It could rewrite the architecture of your brain like a vandal. It could slowly, methodically hollow out your mind, leaving behind a microscopic wasteland, and eventually it would kill you. And it would do all of this, leaving us with a question that hits with the crushing weight of a falling elevator. What if infection doesn't actually need genes? What if a disease isn't driven by a living organism, but by a shape? Because here is the quiet assumption we all make, almost without noticing. We believe that information, in biology, always means a sequence, a code, a text. We believe the human body is defended against invaders that carry readable messages, the chromosomes of a bacterium, the RNA of a virus, the DNA of a parasite. We firmly believe that to replicate, you must copy letters. But think about your own life right now. Your identity, your childhood memories, your distinct sense of me, that doesn't feel like a string of letters, does it? It feels like a pattern, a structure, a living, breathing architecture that holds itself together, right up until the exact moment it doesn't. So tonight, in this lab, holding this material that outright refuses to play by the established rules of reality, we are going to audit a mystery that was once considered absolute scientific heresy. We are going to figure out how a simple protein, just inanimate matter, can behave like a microscopic zombie replicator. What exactly is it copying, if it has no sequence to copy? And how did a series of baffling, horrifying patient cases, a tragic epidemic in a remote jungle community, and a terrifying global livestock crisis force the scientific establishment to completely rewrite the definition of the word pathogen? Come with me. Do not just sit there as an audience member. Step into the shoes of a fellow investigator. Because the only way to truly understand this biological nightmare is to feel exactly how impossible it seemed, right up until the terrifying moment it wasn't. Let us start by stripping away the complexity and looking at the mental model most of us inherit long before we ever step foot in a high school biology class. Imagine life as a massive, ancient library. In this library, DNA acts as the master bookshelves, billions of books written in a simple alphabet of 4 letters, A, C, T, and G. Your cells are the librarians. They read these books, they painstakingly copy them, and they use the instructions to build you. But sometimes, a foreign book slips into the library. A virus, a bacterium, a parasite. That is what we call an infection. A hostile text enters your library and physically hijacks the librarians, forcing them at gunpoint to make thousands of copies of the invader's book instead of your own. That concept is known as the central dogma of molecular biology. It was formulated by Francis Crick in 1958, and it states a simple, directional flow of information. DNA makes RNA, and RNA makes protein. For decades, this flow of information felt as solid, as undeniable, and as universal as the law of gravity. If you have ever pictured a virus in your mind, you have probably pictured a geometric capsule carrying a payload of genetic material, exactly like a seed carrying its hidden instructions inside. So if I stand here and tell you that there is an infectious agent out there with absolutely no DNA and no RNA, your mind does exactly what a rational, educated mind should do. It protests. It fiercely rebels. It says, wait a minute. If it has no genes, then it cannot replicate. It cannot evolve. It cannot have different strains. It cannot adapt. It cannot possibly cause outbreaks. And it definitely cannot start an epidemic. And I want you to know that for years, the world's brightest scientists protested too. Because their protest wasn't emotional. It was entirely rational. Biology had strict rules. Microbiology had strict rules. Epidemiology had strict rules. And claiming you found a genome-free infectious agent sounded exactly like a lunatic claiming you could put a blank, white piece of paper into a photocopier, press start, and somehow print out a copy of Shakespeare's Hamlet. It was absurd. But to solve this impossible crime, we need to zoom all the way down from the grand library to the microscopic machinery on the factory floor. Because DNA is not the organism. DNA is just the blueprint. DNA is not the neuron firing a thought in your brain, it is not the heart muscle contracting to pump your blood, and it is not the immune cell ruthlessly chasing down an intruder. DNA is merely potential. Proteins, on the other hand, are action. Proteins are the physical machines that do the heavy lifting of life. They are built from smaller building blocks called amino acids. I want you to picture these amino acids as beads on a string. A long, flexible chain. A sequence. But here is the ultimate trick of the universe, and it is arguably one of the most important magic tricks in all of nature. A protein's job is not determined solely by the order of those beads. Its job is determined entirely by how that string of beads physically folds itself into a three-dimensional shape. If DNA is a recipe written in ink, a protein is the sharp, steel knife you hold in your hand. And the knife's cutting power doesn't come from the list of ingredients used to forge the steel, it comes from the precise geometry of the blade, the curve of the handle, the exact microscopic angle of the edge. In your body right now, enzymes work because their three-dimensional surfaces cradle other molecules perfectly, like a bespoke leather glove. Receptors on your cells work because their distinct shapes match incoming chemical signals like a secret handshake. Cellular messages travel from your toes to your brain because proteins literally change their physical shape, like microscopic gears shifting in a Swiss watch. Biological life runs on protein folding the exact same way human civilization runs on physical infrastructure. So, here is the clean, terrifying definition, and I want this to land with absolute clarity in your mind. A prion is a misfolded version of a completely normal protein. In mammals, including you and me, the famous one is simply called the prion protein, often abbreviated as PRP. Your body is making it right now as you watch this video. It sits perfectly on the surfaces of your cells, especially the delicate neurons in your brain and spinal cord. Most of the time, it folds into a harmless, elegant, functional shape. It does its job, it degrades, and it is replaced. Harmless. But in prion disease, a version of that exact same protein folds into a different, dangerous, twisted conformation. A different 3D architecture. And that new shape, just the shape itself, is the infectious agent. Not a virus. Not a bacterium. Not a fungus. Not a microscopic parasite. Not a hidden piece of genetic code. Just a shape. Take a second to let that sink in. If that concept feels deeply unsettling to you, good. It absolutely should. Because it means the ultimate threat isn't a foreign invader breaching your castle walls with a new set of instructions. The threat is a corrupt, twisted form of something you already own. It is exactly like discovering that the saboteur didn't sneak into your factory at night. Instead, your own factory machines spontaneously twisted themselves into deadly weapons. But this brings us to a massive, glaring question. A question that makes prion either a trivial biochemical quirk or a terrifying apocalyptic threat, depending entirely on the answer. What does the word, infectious, actually mean in this context? How on earth does a mere shape spread from one cell to another? I want you to picture a flat, pristine sheet of origami paper. You can fold it into a paper airplane, or a delicate crane, or you can crush it into a tight, useless cube. It is the exact same paper. The exact same atoms. But it has a completely different architecture. Now, imagine you have one specific piece of paper folded into a very strange, highly stable, jagged shape. And this bizarre shape possesses a terrifying physical property. Whenever it touches a normal, flat sheet of paper, it exerts a force that physically compels that flat sheet to instantly refold itself into the exact same jagged shape. It doesn't do this by magic. It does this by the laws of physics. By surfaces meeting surfaces. By thermodynamic energetics. By the quiet, invisible force of molecules constantly seeking the most stable energetic state. Biologists call this terrifying phenomenon templated misfolding. The misfolded prion protein acts exactly like an iron stamp pressing down into soft wax. The wax isn't given new chemical ink. It is brutally forced into a new geometry. Inside your brain, if a misfolded PRP molecule bumps into a normal, healthy PRP molecule, it induces that healthy protein to flip, to twist, to refold into the pathogenic, disease-causing conformation. Now you have 2 misfolded proteins. They bump into 2 more. Now you have 4. Then 8. 16. 32. It is a microscopic chain reaction. Then, these newly twisted proteins stick together. They are highly adhesive. They form massive, indestructible aggregates, clumps, long sticky fibrils, impenetrable amyloid structures. Think of it like a catastrophic traffic jam on a massive highway network, a jam that constantly grows by aggressively recruiting more and more innocent cars into the pileup. And human cells, especially the delicate, beautiful neurons in your brain, are exquisitely sensitive to traffic jams. Neurons are incredibly long, fragile, electrically active structures. They absolutely depend on seamless intracellular transport, on perfectly clean cell membranes, and on tightly controlled chemistry. When these misfolded prion proteins start accumulating, the cell's internal quality control systems, tiny machines called chaperones and proteasomes, try desperately to clear the garbage. But they can't. The prion are too tough. The system strains under the massive molecular load. Cellular stress pathways ignite like alarm bells. Outer membranes begin to physically deform. Vital synapses fail to fire. Slowly, inevitably, the intricate architecture of the human brain begins to literally crumble from the inside out, leaving behind gaping microscopic holes. It turns the brain into a sponge. If you have ever had the misfortune of watching someone you love slowly disappear behind the thick, suffocating fog of standard neurodegeneration, like Alzheimer's or Parkinson's, you already know the profound human tragedy of protein misfolding. But prion diseases do not just slowly blur a person's memory over decades. They can absolutely shred a human mind with breathtaking, violent speed. Now, if we are being completely honest with ourselves, the word prion doesn't usually enter a person's vocabulary through a sterile biology lecture. It arrives through sheer terror. It arrives through panic newspaper headlines screaming about mad cow disease. It arrives through whispered stories of sudden, rapid dementia, of terrifying uncontrollable body movements, of a medical helplessness so profound it feels medieval. So, we need to step out of the laboratory and walk through the actual human evidence, the horrifying historical outbreaks, that violently dragged the stubborn scientific community kicking and screaming into this new reality. Let's go back in time to the mid-20th century, deep in the remote eastern highlands of Papua New Guinea. There is an isolated tribe known as the Four People. Imagine being a young clinician or an anthropologist, like Shirley Lindenbaum or Carlton Gajdusek, arriving in these lush, incredibly remote villages, only to find families completely haunted by a strange, horrific illness they called Kuru, which translates to shivering or trembling. People, mostly women and children, would suddenly develop severe tremors. They would lose their physical coordination, stumbling as if intoxicated. Then, a deeply disturbing symptom would emerge. They would begin to laugh uncontrollably. Not because they found anything amusing, but because the delicate neurological circuits in their brains that controlled emotion and facial expression were literally disintegrating. Once the symptoms began, death was an absolute, unavoidable certainty, usually within months. The local people believed it was sorcery. But the scientists mapping the outbreak noticed a pattern. The transmission route, painstakingly pieced together over years of anthropological detective work, was tied to a deeply held mortuary practice, transumption, or funerary cannibalism. It was a cultural ritual of consuming deceased relatives as a profound act of respect, mourning, and keeping the spirit of the dead close to the family. In their culture, it was an ultimate expression of love through tradition. But biologically, it was a perfect, catastrophic conduit for a lethal pathogen that heavily concentrated in brain and nervous tissue. When the women prepared the bodies, and when the community consumed the infected tissue, the invisible agent found new hosts. When the Australian government and missionaries successfully campaigned to stop the practice in the late 1950s, the Kuru epidemic faded. But it faded agonizingly slowly, because scientists realized this invisible killer could incubate inside a human body in complete silence for up to 50 years before striking. Kuru was a heartbreaking, devastating tragedy. But scientifically, it provided one of the clearest, most undeniable epidemiological clues in history that something highly transmissible, something infectious, was destroying these brains. But Kuru wasn't the only ghost haunting the neurological world. Far away from the jungles of New Guinea, in modern Western hospitals, there was Creutzfeldt-Jakob disease, or CJD. It was incredibly rare, utterly devastating, and usually sporadic, meaning it struck entirely at random, as if an invisible bolt of lightning hit a single human brain without warning, affecting roughly 1 in a million people worldwide. Scientists discovered that some forms of CJD were inherited, directly linked to genetic mutations in the PRP gene. This proved that even a microscopic, single-letter change in the protein's natural tendency to fold could tilt an entire human life toward rapid catastrophe. But far more terrifying were the iatrogenic cases. Iatrogenic means the disease was accidentally transmitted through modern medical procedures. Patients were contracting CJD from contaminated surgical instruments used in neurosurgery, or from human growth hormone harvested from the pituitary glands of cadavers, or from corneal transplants. I want you to pause and really think about that. Surgical instruments. Hardened stainless steel. The kind of equipment modern hospitals confidently sterilize every single day. We are entirely accustomed to thinking of hospital sterilization as a universal, invincible reset button. You blast it with high heat in an autoclave, you irradiate it with harsh UV light, you soak it in flesh-burning chemicals like formaldehyde, and the problem is solved. The bacteria pop. The viruses shred. But prion are not living cells with membranes that you can easily rupture. And they are absolutely not viruses with fragile genetic payloads that you can shred with radiation. They are impossibly stubborn, armor-plated proteins locked in a hardened, almost crystal-like aggregated state. That is precisely why standard medical sterilization utterly struggles against them. Not because prion are magical or supernatural, but because they are chemically and thermodynamically resilient in ways that standard, nucleic acid-based pathogens simply aren't. High heat that would easily melt a bacterium might not fully denature a dense prion aggregate. UV radiation that aggressively destroys the DNA of a virus does absolutely nothing to a prion, because the prion has no DNA to destroy. Many harsh chemical disinfectants that dissolve viral lipid envelopes cannot penetrate a dense amyloid fibril. To properly destroy a prion on a surgical scalpel, hospitals have to use extreme, almost destructive protocols, like soaking the steel in highly concentrated sodium hydroxide, Li, followed by extended autoclaving at extremely high pressures and temperatures. These specialized decontamination protocols exist for one very uncomfortable reason: the medical establishment's usual weapons simply bounce off. And then, of course, the world experienced the true, visceral horror of the prion paradigm, BSC, or bovine spongiform encephalopathy. You know it by its media moniker, mad cow disease. Picture the United Kingdom in the late 1980s and early 1990s. Farmers began reporting cattle displaying highly erratic, strange behavior. Massive, 1,000-pound animals losing their coordination, becoming irrationally fearful, collapsing, and showing terrifying aggression. Farmers watched helplessly as herds they had raised for generations became completely unrecognizable. Suddenly, an entire nation's food supply chain was under deep, existential suspicion. Epidemiologists frantically traced the source. The most widely accepted explanation pointed directly to modern industrial agricultural practices. To maximize protein intake and efficiency, cattle were being fed meat and bone meal, processed biological remains of other cattle and sheep. It was a forced biological loop. A massive, industrial-scale system designed purely for economic efficiency had accidentally become a countrywide prion amplifier. The government insisted beef was safe. Politicians ate burgers on television to calm the panicked public. But then, the unthinkable happened. A new, terrifying illness emerged in young, otherwise healthy humans. It was called variant CJD. It was directly linked to human exposure to BSE-contaminated beef products. The abstract scientific puzzle was no longer abstract. The barrier between species, The biological moat we always like to imagine kept us perfectly safe from animal diseases suddenly looked a lot less like a fortress wall, and much more like a broken chain-link fence with massive gaps. People realized that a burger they ate a decade ago could currently be ticking like a silent, microscopic time bomb in their brain. The terror was absolute. At this exact point in our investigation, a highly logical question should be screaming in your mind: if prion are just proteins, why did it take the scientific community so incredibly long to accept them as infectious agents? Why wasn't this undeniably obvious the moment someone first proposed it? Because the proposal didn't just add a quirky new microbe to the textbooks. It threatened to burn down the entire bookkeeping system of modern biology. The central dogma was sacred. Infectious agents were legally required to carry genetic instructions. If you remove the instructions, you entirely remove the mechanism for replication. So, I want you to imagine you are in the lab with Dr. Stanley Prisoner in the early 1980s. I don't want you to look at this in hindsight, when Wikipedia and modern textbooks make his Nobel Prize-winning discovery sound smooth and inevitable. I want you in the thick of the blinding confusion and the bitter, academic vitriol. You run experiment after grueling experiment trying to find the hidden virus, the hidden nucleic acid causing these diseases. You take infectious brain mash from scrapey infected hamsters. You aggressively treat the samples with heavy radiation meant to completely obliterate DNA and RNA. You add powerful nucleases, molecular scissors that chew up genetic material into useless dust. You use rigorous purification protocols that physically separate proteins from nucleic acids. Every single time you do this, you fully expect the infectivity of the sample to collapse to zero. You inject it into a healthy animal, expecting the animal to live. But it doesn't. The disease transmits. The animal dies. The incubation curves remain perfectly, terrifyingly unchanged. It is exactly like hunting a ghost, cornering it, shooting it with ghost-killing weapons, and watching the weapons pass right through it. That is the exact moment where science ceases to be a sterile profession and becomes a brutal psychological trial. Because when your data contradicts reality, you can either assume your methods are deeply flawed, which your colleagues will loudly tell you, or you can assume the current model of human reality is fundamentally incomplete. Choosing the latter is what separates routine benchwork from the bleeding edge of a genuine scientific frontier. Dr. Prisoner coined the term prion, short for proteinaceous infectious particle. For years he was mocked. He was called a heretic. The scientific backlash was fierce, sometimes deeply personal. But eventually, the sheer weight of the physical evidence corners you. Not with one dramatic, cinematic, eureka moment, but with a slow, grinding convergence. Multiple, independent lines of data, all relentlessly pointing to the exact same, completely impossible conclusion: the infectious agent behaves entirely like a protein. The infectious property perfectly tracks with a specific misfolded conformation. And no detectable nucleic acid is required. And now, my fellow investigators, we have finally arrived at paradox number 1, replication without copying code. How in the name of physics can something replicate if it does not encode the instructions for its own manufacture? The ultimate key to unlocking this paradox is to realize that the prion isn't actually manufacturing new protein from scratch. It isn't building anything. The host cell is already manufacturing healthy PRP as part of its daily, normal biological routine. The invading prion doesn't need a blueprint to build the raw material. The raw material is already floating around by the millions. The prion only needs the physical power to reshape what already exists. So, biological replication here is not like printing a brand new book at a printing press. It is like breaking into a library, taking existing, perfectly good books off the shelf, and brutally forcing them all to fold into the same weaponized paper sculpture. It uses the exact same pages, the exact same ink, just a completely different, deadly form. And once you finally accept that mechanism, a much deeper, much stranger philosophical implication arrives at your doorstep. The information, being copied from host to host, is not a sequence of biological letters. It is a stable geometry. In the realm of quantum physics and materials science, we sometimes talk about the concept of state. A physical system can exist in radically different states even if its fundamental components are 100% identical. A piece of iron can act as a magnet pointing north, or it can point south. A carbon material can be a soft, smudgeable graphite pencil lead, or it can be a perfectly rigid, indestructible diamond. Water can be a solid block of ice, a flowing liquid, or an invisible vapor. It is the exact same molecules, just a different physical organization. An organization is not just a poetic descriptor. Organization is a hard, physical reality that changes everything about how the object interacts with the universe. Prion forced the field of biology to humbly admit the exact same truth. Two proteins with the exact, 100% identical amino acid sequences can carry wildly different biological identities simply because they occupy different conformational states. In the world of proteins, the fold is not merely a decoration. The fold is absolute destiny. But just as you wrap your head around that, paradox number 2 hits you like a freight train: strain diversity without genes. In the world of standard viruses and bacteria, different strains exist, like the Delta or Omicron variants of a virus, because their genetic codes literally differ. Mutations occur, genes recombine, natural selection picks the winners. But if prion have absolutely no genome to mutate, how on earth can there be distinct prion strains that have completely different incubation times, that target completely different regions of the brain, and that produce completely different clinical symptoms in patients? The answer is both incredibly elegant and deeply unsettling. There isn't just one single misfolded shape that the normal PRP protein can fall into. The energy landscape of protein folding is incredibly complex. There can be multiple, distinct, stable misfolded conformations, essentially, different ways of being wrong. Each unique, twisted conformation acts as its own specific template. It forces the normal proteins to adopt its exact specific geometry, propagating its own unique shape like a molecular signature. So, instead of a traditional evolutionary family tree written in genetic A's, C's, T's, and G's, you get a sprawling, three-dimensional landscape of shapes, peaks and valleys in the microscopic energy terrain. Each valley represents a different stable fold. Each fold acts as a different strain. Darwinian selection still happens, but it is not a selection of genes. It is a ruthless physical selection among competing conformations, among microscopic architectures violently competing for raw protein material and cellular environments. If you want a metaphor that doesn't lie to you, imagine a massive crowd of people doing a complex dance in a stadium. The people are the exact same. The music playing over the speakers is the exact same. But if one specific, strange choreography catches on in a corner of the stadium, it spreads outward purely by physical imitation. Not because anyone is carrying a written instruction manual, but simply because the physical pattern is visible, it is stable, and it is highly contagious. Now, here is the ultimate thought experiment. I want you to hold this tightly in your mind, because it thoroughly stress tests exactly how you think reality stores the concept of identity. Imagine I place 2 glass test tubes on the bench in front of you. In both tubes is the exact, identical protein sequence. Atom for atom, bead for bead, electron for electron, they are identical. In tube A, the protein is folded normally. In tube B, it has slipped into a pathogenic prion conformation. If you rigidly clung to the old central dogma, if you only believed that identity lived exclusively in genetic sequence, you would look at those tubes and declare that A and B are the exact same thing. But if you inject those tubes into a living, breathing biological system, the outcomes are radically, terrifyingly different. One tube is completely inert, it does nothing. The other tube seeds an unstoppable, terminal cascade that destroys a mind. So I ask you, which tube actually contains the agent? In this strange, shadowy realm of prion biology, the undeniable answer is tube B. Not because it contains an ounce of new biological matter, but because it contains a different form of physical information. And recognizing that requires a massive category shift in human thought. This is exactly where I want to slow down. We've been moving incredibly fast through complex biochemistry and high-level mechanisms, And it is very easy for the profound human meaning of this to get lost in the academic jargon. Your brain is not only a bag of chemistry. It is an architecture. It is a living, breathing cathedral of billions of connections, painstakingly built from cells that constantly grow, retract, signal, and remember. Your deepest memories, your first kiss, the smell of your childhood home, the sound of your mother's voice, are not stored in a static DNA paragraph that can be easily read out like a book on a shelf. They are dynamically stored in the physical strengths of your synapses, the complex rhythmic patterns of neurofiring, the precise distribution of protein receptors. Your mind is a structure shaped by time and experience. If a physical architecture can be infected, then disease is no longer just a story about foreign invasion. Disease becomes a story about structural deformation. It becomes about forced, molecular origami, innocent molecules violently coerced into a shape that relentlessly spreads its own coercion. And once you finally see that logic, once you put on those glasses, you start noticing this prion-like logic hiding elsewhere in the biological universe. It's not always an infection, it's not always a terrifying public health hazard, but it is a fundamental, underlying principle of life. Out in nature, we are discovering functional amyloids, protein aggregates that organisms actually use on purpose, as tools. Certain bacteria deliberately form tough biofilms using amyloid fibers acting as structural scaffolding. Some animals utilize amyloid-like structures to safely store melanin pigmentation. Biology, it turns out, doesn't always fear aggregates the way our brains do. Over millions of years of evolution, it has sometimes learned to recruit them as tough, resilient building materials. Inside your own cells right now, proteins can undergo a process called phase separation. They form distinct, liquid-like droplets without needing lipid membranes, organizing complex biochemistry just by gathering certain molecules together in a dense crowd. It is exactly like oil droplets separating in a glass of water, but with the staggering complexity of living chemistry. These are absolutely not disease-causing prion, but they remind us of a profound truth: physical state, exactly how molecules dynamically assemble and arrange themselves, can carry deep biological meaning. And then, we arrive at the highly controversial, bleeding-edge frontier that people always inevitably bring up: what about the other giants of neurodegeneration? What about Alzheimer's disease? Parkinson's? ALS? Huntington's? All of these devastating diseases fundamentally involve misfolded proteins and the slow, relentless buildup of toxic aggregates in the brain, amyloid beta, tau, alpha-synuclein. And in highly controlled experimental laboratory settings, researchers have observed clear, seeding behavior. They inject a tiny seed of an Alzheimer's aggregate into a mouse model, and it encourages more aggregation to rapidly spread through the brain tissue. Now, we need to be incredibly rigorous and responsible here. I am not saying these diseases are exactly the same. Not all neurodegenerative diseases are infectious in the real world like prion diseases. The epidemiology is vastly different, the real-world transmission risk is not the same, and the public health implications should absolutely never be sensationalized. You cannot catch Alzheimer's from sitting next to someone on a bus. But the deeply shared underlying logic, the misfolding, the templated seeding, the slow, domino-like propagation of a toxic shape through brain tissue, strongly suggests that true prion are not just a bizarre, isolated freak of nature. Prion may actually be the sharpest, most terrifyingly clear example of a much broader, universal physical principle. In biology, structure can self-propagate. So, as we stand here at the end of this audit, what does all of this actually do to our worldview? Here is the profound philosophical takeaway that slowly sneaks up on you if you spend enough late nights staring into this topic. Biological life is not exclusively written in sequential codes. Life is also sculpted in physical forms. Information can be purely physical. Not metaphorically physical, literally physical, deeply embedded in three-dimensional geometry, in spatial arrangement, in the perfect fold of a microscopic chain. Since the 1950s, we have been rigidly conditioned to think that DNA is the undisputed master of the cell, the absolute sovereign, the sacred, untouchable text of life. But DNA is far more like a vast, silent library that cannot lift a single finger to change the world. Proteins are the actual, physical machines that construct and move the world. And their diverse, intricate shapes act as the ultimate operating system of life, strictly governing what biological actions are possible, what chemical interactions can occur, and what cellular pathways open or close. If that analogy sounds slightly odd to you, take a moment to notice exactly what it buys us in understanding. It perfectly explains why a single, microscopic genetic mutation can result in wildly different medical outcomes for a patient, depending entirely on the folding environment of the cell. It explains exactly why changes in body temperature, pH levels, cellular stress, and the availability of chaperone proteins can dramatically tip a healthy biological system over the edge into catastrophe. It perfectly explains why having the same sequence does not inherently guarantee the same function. And ultimately, it forces upon us a deep, profound ethical and ecological humility. The world can and does host deadly hazards that are definitively not alive, yet they behave with the fierce, replicating drive of the living. Prion do not metabolize energy. They do not reproduce by splitting a cell in two. They do not consciously hunt for a host. They absolutely do not think. And yet, against all logic, they propagate, they stubbornly persist across decades, and they have the raw power to fundamentally reshape agricultural ecosystems and terrify human societies. Our strict human definitions, words like living, pathogen, infection, and agent, are ultimately just comfortable conveniences. They are boxes we built to make us feel like we understand the universe. But nature doesn't care at all what words we prefer to use. Nature only cares about what physically works. There is also a cold, sweeping sense of awe hiding in this science, the kind of awe that makes you stare down at your own two hands as if you are seeing them for the very first time. Prion revealed to us a hidden universe where pure geometry can spread like a forest fire. No genes required. No intention needed. Just the raw, undeniable logic of shape matching, of rigid templates brutally recruiting raw material, of physical patterns ruthlessly preserving themselves by converting the world around them into more of the pattern. And now, we can finally close the loop and return back to that late-night moment in the laboratory, the gloved hands, the humming fluorescent lights, and the frosted vial that supposedly shouldn't be able to frighten us. Because the true terror isn't only that these genome-free, zombie proteins exist in the world. The deeper terror, and perhaps, if you look closely enough, the deeper, staggering beauty of it all, is what their existence inherently implies about you and me. If the concept of the self is essentially a pattern, if a human mind is nothing more than a breathtakingly fragile architecture built from billions of proteins folded just so, with synaptic connections precisely tuned just so, then protecting human life means doing much more than just protecting our cells and guarding our genetic codes. It means we must desperately protect the incredibly fragile, microscopic forms and geometries that allow a mind to be a mind. Let us synthesize exactly what we have dragged into the light on this journey. We began with the deeply held, dogmatic assumption that all biological infection strictly requires a genetic code. We then relentlessly followed the historical and clinical evidence into a terrifying new category of disease: prion, twisted, misfolded proteins whose sole infectious property is their three-dimensional conformation. We audited the exact mechanics of templated misfolding, visualizing how a microscopic shape can replicate by brutally converting normal, healthy proteins into the exact same pathogenic fold. We confronted the mind-bending paradox of how strain-like diversity can easily emerge from multiple, stable misfolded conformations without a single mutation in a gene. And ultimately, we recognized that prion completely shatter and expand biology's narrow definition of information, proving that profound, world-altering data can live entirely within geometry. Now I want to leave you with a concrete, physical takeaway that you can carry into your own life, something tangible you can test against your own intuition today. Tonight, or tomorrow morning, I want you to pick up an ordinary, physical object in your home. It could be a simple metal paperclip, a house key, or a silver spoon. Hold it in your hand and sincerely ask yourself, how much of this object's true identity is bound in the raw material it is made of, and how much of its identity is bound strictly in its physical form? If you take a blowtorch and melt that spoon down into a shiny puddle, you absolutely still have the exact same atoms of metal, but you no longer have a spoon. Its identity is gone. In a much quieter, infinitely more complex way, You are exactly like that metal object. Your body completely replaces nearly all of its constituent atoms over the course of a few years, yet you steadfastly remain you. Why? Because the continuity of your life is not only anchored in chemistry. It is anchored in structure. It is anchored in organization. And that realization brings us right to the threshold of the next great, towering question. The question that slowly opens the next heavy door in our audit of the universe. If microscopic geometry can carry biological information strongly enough to behave exactly like a deadly pathogen, what other incredible, entirely unseen kinds of information are currently hiding in the vast shapes of the world? What mysteries are folded up inside our cells, inside the deep architecture of our brains, or perhaps even inside the fundamental way we physically experience the flow of time itself? If you want to keep going down this rabbit hole, I've put a video on the screen right now that takes this precise idea of information existing beyond code and pushes it into an even stranger, much deeper place. Click it, and let's continue the audit while the thread of curiosity is still hot in your mind. And before you go, I need to hear from you down in the comments, because this specific topic always perfectly reveals what people intuitively believe about the nature of life. Be honest, before watching this video, would you have confidently said that biological information absolutely must mean DNA or RNA, yes or no? Answer with just that one word, and if you have a moment to add a sentence, tell me exactly what part of this audit changed your mind. Finally, if you are not already subscribed to the channel, click subscribe. Don't do it just for more random trivia facts. Do it for more deep audits exactly like this one, where we take one seemingly impossible claim, we ruthlessly follow the evidence without ever flinching, and we come out the other side with a vastly deeper, clearer model of reality. Stay curious, and I will see you in the next audit.